Exopolysaccharides for probiotic fortification and food processing, a technology developed by Dr. Madhavan Nampoothiri from CSIR NIIST. Exopolysaccharides are extracellular macromolecules produced by food-grade bacteria and used as a source of polysaccharides. Polysaccharides are widely used in food industry as additives to enhance flavor and texture, prevent syneresis, improve viscosity, sensory appeal, and shelf life of food products. Polysaccharides are also used in cosmetic industry as rheology modifiers and making facial masks. In pharma, they are used as API as well as excipient for stability and targeted drug delivery. Food packaging industries use polysaccharides for their biodegradable thin film forming ability. EPS from lactic acid bacteria have US FDA GRAS status and are widely used for processing of raw foods, industrial fermentation, development of prebiotics, which are food for beneficial microorganisms in the body, as well as postbiotics, which are metabolites or healthy byproducts produced by these microorganisms as part of their life process. Market opportunity. The global market for polysaccharides was valued at USD 14.04 billion in 2021 and is expected to reach USD 21.41 billion in 2030, growing at a cumulative annual growth rate of 4.8% during the forecast period. Rising awareness for clean label products with unadulterated and organic ingredients is creating a demand for naturally produced polysaccharides. With development of newer manufacturing techniques, there is an increasing use of polysaccharides in animal feed for nutritional benefits as well. Market leaders in this segment include DuPont, Cargill, Tate & Lyle, Dow Chemicals, etc. Some of the challenges faced by this segment include optimizing cost while increasing yield of production. EPS also finds use as postbiotics, and the global market for postbiotics is expected to grow at a cumulative annual growth rate of 6.8%, reaching a value of USD 3 billion in 2031 from a value of USD 1.6 billion in 2021. A steep market growth for postbiotics is foreseen in personal care and cosmetics industry, followed by food and beverages industries. Who should be interested in this technology? Manufacturers of functional foods with focus on health and nutrition, where there is scope for novel postbiotic products, and developing products with increased stability and shelf life. Manufacturers of food ingredients, dairy products, and frozen foods like plant-based yogurts and ice creams can also utilize the anti-syneresis property of EPS for creating novel products with enhanced nutritional benefits as well as increased stability and shelf life. Cosmeceutical and pharmaceutical companies can also benefit from this natural source of polysaccharides for various applications including rheology modification, developing delivery systems, etc. About the technology. The process features microbial synthesis of exopolysaccharides using food-grade bacteria of both wild type as well as genetically improved strains. The process has been optimized for high yield of EPS using relatively low-cost raw materials. Raw materials with high starch content such as cassava starch hydrolysate, jackfruit seeds, with relatively lower costs have been explored. The EPS produced is secreted out into the broth, providing advantage of ease of extraction in downstream processing. The final product is of high quality and powder consistency. Two types of EPS have been produced: homopolysaccharides like dextran EPS, having high molecular weight with good anti-syneresis properties. Dextran EPS has reported highest yield of 19.92 grams per liter. Heteropolysaccharides like glucomannan EPS were also produced, having known properties such as antioxidant property and cholesterol reduction properties. In vitro cell line studies were carried out showing potential anti-tumor activity as well. A case study for testing anti-syneresis activity, that is, prevention of separating out of water layer from yogurts using dextran EPS, was carried out, and this activity was compared with other stabilizing agents such as sodium alginate, pectin, carboxymethylcellulose, cornstarch, etc. Dextran EPS at very low concentration of 0.2% showed significant anti-syneresis activity. Yogurt with EPS also had higher antioxidant properties than control yogurt. This was tested periodically during 21 days of storage using DPPH assay for measuring the radical scavenging activity of antioxidants. Addition of EPS also gave better mouthfeel, texture, color, as well as creaminess to the yogurts, as concluded by sensory and texture evaluation by test subjects. Current status. The technology is demonstrated at lab scale using 5-liter fermenter capacity, and the patent application filing is in progress. About the team and organization. CSIR National Institute for Interdisciplinary Science and Technology (NIIST) is a constituent laboratory of CSIR engaged in research and development activities in the field of agro-processing and technology, chemical sciences, material sciences, microbial processes, and environmental technology. The lead scientist, Dr. Madhavan, is presently working as Chief Scientist and Head of the Microbial Processes and Technology Division of CSIR NIIST. Dr. Madhavan is actively involved in microbial biotechnology research with special emphasis on bioprocess technology and product development for the last 28 years. His significant contributions in the area of industrial enzymes, microbial metabolites, genomics and infectious diseases, probiotics and nutraceuticals, etc. His scientific credibility includes 7 patents, nearly 200 research review papers, over 140 research communications in international conferences and national conferences, and around 45 NCBI gene submissions. His team has a total of 14 people. CSIR NIIST has well-developed labs and facilities, including fermenters ranging from 2 to 150 liters, a pilot plant facility for bioethanol program, 2G ethanol biorefinery technology, biomass valorization for fuel and chemicals with daily processing capacity of 80 kilograms, Koji room for large-scale solid-state fermentation facility, and 50 to 80 kgs of biomass holding trays. Various analytical systems such as HPLC, GC, LCMS, gene sequencer, ultracentrifuge, amino acid analyzer, gel permeation, and flash chromatography are available. Dr. Madhavan is coordinating several CSIR network projects in NIIST and many other funded projects from DBT, DST, and has international research collaborations with institutes and universities in Germany, France, UK, Mexico, and Portugal. He is a member of 2 technologies transferred from NIIST on microbial enzymes. Next steps. The team has developed the background science and demonstrated EPS production at lab scale 5-liter fermenter using wild type as well as genetic improved strains. Further work have been done on developing and testing products with beneficial properties, using EPS as well as natural cultures for use in dairy products, yogurts from various plant-derived milk, etc. The team has expertise to modify and get desired products. Next phase will be to work closely with industry partners to optimize process and product as per their requirements. The scientist is seeking industrial partners for technology licensing or for sponsoring further technology advancement and scale up, or even explore opportunities for raising third-party funds for a collaborative project. Interested industries can reach out for tapping scientist capabilities as an expert or consultant. For more information, please contact Case Manager Pradnya Aradhye. Thank you.